Hello, am I audible now? Can someone just type in the chat if I'm audible? Wonderful. Thank you very much for being here, and we apologize greatly for making you guys wait because our speaker was supposed to be Aisha Haq, but she has been caught up in an emergency, unfortunately, and she couldn't make it to today. So I'm going to take over so we don't waste your time anymore. So, without any further ado, let's talk about our today's topic. Our topic of the today's session is sweet science. Why do we call it sweet science? Because it is detecting sugars with test. Benedict test is a test in chemistry that is really known in school. We perform experiments with this, but we never have ever do a hands-on workshop or practice this. So we are going to talk about the practical applications of Benedict test, a hands-on workshop on reducing sugars and enzyme action. This event is in collaboration with Project Q2, Youth Stream Advisors, and Harvard Undergraduate Microbiology Society, and we're excited to proceed with you guys. So today we are learning a fundamental technique in biochemistry and microbiology. And just to give you a little bit of overview for anyone who's new here and does not know what is a Benedict test, we are going to understand today what reducing sugars are and why do they matter. And we learned how to perform and interpret a Benedict test, investigate how the concentration of reducing sugar affects the reaction time. Yeah, that is very interesting how the concentration of a sugar can affect your reaction time. It can affect how the color changes, and that's what we are going to observe in our today's workshop. And let's get ready for our short hands-on lab session. What is the science behind the test? And what are reducing sugar, what are Benedict region, and what is the reaction? Let's talk about everything. If you can grab sugar and the ingredients and the tools needed for the experiment so we can observe it more practically. And you can put down any questions in the chat box if you want. Reducing sugar is sugar with free aldehyde or ketone group. Example, glucose, fructose, and they all act as reducing agent. So what are reducing agent? Reducing agent are agents that reduce other particles or other sugars. And the most famous examples and the one we study mostly in our books are glucose and fructose. Then we have Benedict's region. We'll take Benedict region, which is an alkaline solution of copper 2 sulfate, and it is bright blue in color. If you can observe it, you can have a look at the picture. Unfortunately, Aisha was supposed to provide the practical explanation and the overview as well, but since she's not present, there you are going to face a little inconvenience, and we apologize for that. Now let's talk about the reaction. When we heat a reducing sugar, the copper 2 ions are reduced to copper 1 oxide. That's how it is reduced. Then this forms a colored precipitate. So it goes a color change gradually, and it's very fascinating to observe how such simple experiments and reactions can observe different colors and different changes. So it goes from blue to green to yellow to orange and to finally to brick red. So the more sugar we add, the more faster the reaction will be and the color change will be more dramatic. So if you can, you should go and grab a sugar and observe this practical explanation. It is very interesting to have a look at this. Now, let's see how the concentration of affects Benedict test reaction time. Now, if we take the following materials, boiling water bath, test tubes, Benedict solution, and reducing sugar solution and stopwatch, we are going to start this practical explanation. First of all, we'll ensure that our water is boiling. Then we'll have two labeled test tubes for each sugar concentration. And for each test tube, we'll add 2 centimeter cube of Benedict solution, the solution that we just talked about. Then we'll add 2 centimeter cube of our assigned reducing sugar solution to the corresponding test tube, and we'll start with the 0.5% of the concentration. Then we'll place the test tube into boiling water bath, and we'll just, as we mentioned, we'll take a stopwatch and we'll start the timing immediately. We'll watch the solution really carefully and record the exact time in seconds. When you see the first visible color change from blue, as we just talked about, the original color is blue, you'll see the first visible color change within a few seconds. And when, if you do not see any change after 120 seconds, you record as less than 120 seconds. And then you repeat all the steps from 4 to 7 for all the other sugar concentration. So the purpose and the objective of this experiment and this practical is to observe how different sugar concentration affect how the color can be observed, how the color is different, as we just talked about in the previous slide as well, that the color can change from blue all the way to brick red, and that depends on the concentration of the sugar solution. Now, these are some of the expected results. If you choose to perform these experiments, you are going to observe different colors within 120 seconds. You'll go from no change, it will stay blue. This is what's going to happen in the first 120 seconds when you have no sugar. It's just water. When you add 0.1% of sugar, the time will be quite slow for 60 to 90, 90 seconds, and you'll observe a very faint yellow and green color. After then, we'll add more sugar, which is 0.25% concentration, and we'll change, and the color will gradually change to yellow within 30 to 60 seconds. We'll add more sugar now, 0.5% of the concentration, and it will proceed to very fast speed within 15 to 30 seconds, and the color observed will be orange. And now that we add the highest concentration we have right now, which is 1.0% sugar, the reaction time would be quite fast. It would be more than 15, it would be less than 15 seconds, and the color would be brick red. Brick red is a really beautiful color. You should observe that. Unfortunately, we are so sorry that Ash isn't here with us. Otherwise, it would have been an amazing session. But we do have another workshop coming soon with another speaker, and hopefully everything is going to be really fine. So we look forward to that with you. Now, now I want to ask you guys, if you guys have any questions till now, you can drop it in the chat box. Otherwise, we'll proceed to the next slide. You can just say no if you do not have any questions, so we can just proceed. I highly appreciate you guys being present here. It's wonderful to see all these people. Oh, I see we have a question from Rafael. Okay, I'll wait until more of you guys drop more questions and I'll answer this one as well. Really interesting and really good question. I appreciate it. Okay, I see we don't have any more questions, which is fine. I hope guys will have more questions as we proceed. Uh, before that, before we proceed, I'll answer Rafael's question. Can you explain the difference between reducing and non-reducing sugars with a molecular model? So, just to make it quite simple, reducing sugars are sugars that can donate electrons because they have a free aldehyde or ketone group. These are carbohydrate groups, and due to the presence of free aldehyde or ketone group, they allow with solutions like Benedict or Felling solution, which gives them a colored precipitate. As I mentioned, some of the examples like glucose, fructose, and maltose. And even if they're in cyclic form, but they have free aldehyde or ketone group, their rings can open and expose the reactive group. So, for example, we have um, glucose, which is a reducing sugar, and it is a monosaccharide. As you might guys know that carbohydrates have different groups: monosaccharide, oligosaccharide, disaccharide, polysaccharide. So glucose is comes under monosaccharide. It has free aldehyde group in its open chain form, and even when if it's in cyclic form, it exposes to uh, opens to expose the aldehyde, and due to this, it allows to reduce copper ions in the Benedict test. On the other hand, we have non-reducing sugars, which do not have any free aldehyde or ketone because their reactive ends are linked by glycosidic bonds. They do not react with Benedict solution. For example, sucrose, because both the carbons are bonded. So in short, if a sugar can reduce copper ions, these are reducing sugars, and if it cannot, they are non-reducing sugars. I'm trying to find a picture to show you guys regarding that. Let me see. Okay, I'm going to find it at the end of the session. Until then, before we we will continue, and I see youth stream advisors have put the answer for you in the chat box as well. So I hope it sums up the answer. Now let's move to the next slide. Now we are going to apply your knowledge. I hope everything is clear by now, and you know what is a Benedict test, what are reducing sugar, what are non-reducing sugars. Now let's consider a scenario. You have two unlabeled drinks. One is regular soda, which is high in sugar, the one we drink normally, and one is diet soda. It does not have any sugar. Now you perform a Benedict test on both of them. Now, once you start heating them, what would you expect to see in each test tube after heating? That is the first question. If the diet soda turns green after 90 seconds, what might this tell you? I'll let you guys think before we move forward and I tell the answer. So just do drop the answers in the chat box. I'm going to wait. Until then, till you answer, I see Rafael has answered the question, which is really great. I'm going to put the link in the chat box of the um, workshop worksheet so you can fill it in, guys. Everyone can fill it in, and you can give it to us as well if you want with the answers. I'm going to put the link in the chat box. Or maybe if the youth team advisors team could put the link in the chat box, that would be quite better. The one that contained the worksheet. Until then, we'll proceed. Okay, so as Rafael answered, the regular soda turns brick red, whereas the diet soda keeps the same color. Wonderful. So for the first question, as we ask, what would you expect to see in each test tube after heating? As the first test tube, which contains soda high in sugar, it has, as it has sugar. when we perform the Benedict solution in it, it will undergo a color change, which would be high in color change, and it would undergo to brick red. And if the dyed soda turns green after 90 seconds, as the second question asks, it tells that there might be some sugar present, but the concentration of sugar would be really small. As your fail also answered, there's some sugar present in the diet soda, but since it is green and not red, the concentration of sugar in the soda is small, and is quite the correct answer. Meanwhile, the other labeled unlabeled drink, which is the diet soda, and it has no sugar, there would be no change and no color change in that test tube. So, to sum up everything, as the conclusion box states as well, Benedict test is a simple, reliable way to detect reducing sugars. The test works because, as I just mentioned, reducing sugar donate their electrons, and then they change the copper ions in the region. So when we have high sugar concentration, the color change would be faster, and that's what happened in this scenario as well. And the color itself, green, orange, brick red, it gives you an estimate of how much sugar is present. If the color is brick red, it means it has the highest concentration of sugar. So this technique is used a lot in real world applications like urine glucose testing for diabetes and quality control in food production. So hopefully, I hope you guys had a good time hearing it. And I see we do not have the worksheet in the chat box right now, so I'm going to share it first of all. Until then, if anyone of you has any question, please drop it in the chat box. The worksheet is in PDF form. We will send it to you by emails, as we have all of your emails. If you guys have any questions, we have a few minutes left, and you can hop in any questions or any feedback you have. We highly apologize for making you wait. And uh, yes, yes, wonderful. We can send the slides as well. I'll send it by email. And if you have any questions, please do ask. We have a few minutes left, and we have another workshop coming up in March. It might be around 15 March, but we haven't confirmed as well. So we look forward to having a more constructive and more hands-off workshop. Unfortunately, Aisha couldn't join today as she had a really bad emergency. Wonderful. If we do not have any questions, I think we can wrap up the chat. I had a really good time speaking with you guys, and I hope you guys enjoyed as well. Thank you everyone for joining. Let's I'll send the details and the slides to you by email. Let's wrap it up.